Hello everyone, welcome back to Believers Science Academy, Bengaluru. This is topic of the day discussion session. In today's session, we'll be discussing a very important and an interesting topic, that is locust swarming and a new research on pheromone controls or pheromone control. We'll see what is the pheromone control and what does locust swarming mean and what is the significance of this discovery. Okay, so why is it in news? Researchers from the Institute of Zoology, Chinese Academy of Sciences, have identified the pheromone responsible for locust swarming and demonstrated a molecular strategy to block it. The study, which was published in the Nature journal, offers a proof of concept for eco-friendly locust control, a potential alternative to harmful pesticides. So first we'll understand what is this locust swarming. So these are the locusts. Okay, this is the locust, an individual locust, and when these locusts come together in a swarm of number, then it is called as locust swarming. They are a very deadly combination which feeds upon the crops. So the locust swarming or the locust menace is locust and on an individual is not such a big issue, but locusts in swarms are so dangerous that they can consume or eat as much food as 35,000 people per day just per square kilometer. Okay, meaning 35,000 people will go starving if the locust swarm occurs in a particular area. Major swarms occurred in East Africa, Pakistan and India in 2019 and 2020, which was the worst swarm in 25 years. Currently, the control methods heavily rely on pesticides or insecticides. So these are chemical in nature which harm the soil, ecosystems as well as human health. So now this discovery says that it is going to block this locust swarming through molecular technology. We will see what the technology is. So first is, how does this swarming come along? How do they realize where to go, which areas to find, where to feed upon? How do they come together in such a large numbers? So where do the herd mentality come from? The new study says that there is a trigger which they called as swarming trigger. Swarming trigger. The swarming trigger is a 4-vinyl anisole. A 4-vinyl anisole is a pheromone, that is, the locust release 4-VA, which is an aggregation pheromone, which is released after feeding, which draws other locusts and imitates a gregarious behavior or initiates gregarious behavior. What happens here is, so they first it is triggered by serotonin release due to leg rubbing. What happens here is, an individual locust goes and eats something on a crop. Suppose this is corn crop. It goes and eats something here, and there is a serotonin release which is again further caused by due to rubbing of their legs. When they start rubbing their legs, they release 4-VA, that is 4-vinyl anisole. What does this do is, it attracts other swarms. It attracts other swarms to come here and feed, and this attraction and also which is going to lead to gregarious behavior, that is frenzy feeding. So they are attracted by this 4-VA which is released by an individual locust. So then other locusts start releasing as they start rubbing, and as a result so many swarms come together. So what is the role of phenylalanine? So this is an amino acid. What it does is, the amino acid phenylalanine, which is present in plant material, is metabolized by locusts after eating it. So this is actually present in plants. Locusts eat it and metabolize. When it is metabolized, it is converted into 4-VA via 2 key enzymes. So the source is not within the locust, it is within plants. When the locusts eat the plant, they consume this phenylalanine. When this phenylalanine is consumed, it is converted into 4VA. 4VA, that is 4VA, which triggers swarming. How it is converted? Through two enzymes. One is 4V-PMT primary, second is 4V-PMT secondary. So now you understood the reason behind swarming, reason behind the trigger, reason behind how 4VA is released. Now, new discovery says that we can disrupt their 4VA biosynthesis. That 4VA is synthesized when you eat phenylalanine and then it is converted through 4VA PMT 1 and 2. Now the new transaction or new discovery says that the genetic deviation of the 4V-PMT1 gene prevented the transition from solitary to gregarious phase. That is, what the scientists or researchers did is they deactivated this 4VPMT1 gene. It codes for the enzyme 4VPMT, which is the enzyme which results in conversion of phenylalanine into 4VA. Now, if you are able to deactivate this gene, then there will be no VA released. Okay, and using 4-nitrophenyl, that is 4NP, researchers blocked the 4PV-PMT enzyme, stopping 4VA production. How did they stop this? They stopped this genetic deactivation, or they did the genetic deactivation with the help of 4-nitrophenyl. Okay. Now what happens here is when there is no 4VA produced, then there is not going to be any triggered action, then there will be no swarms. So, but there are certain challenges. What are the challenges? This 4NP, that is 4-nitrophenyl, is toxic in nature and environmentally persistent, that it stays up to 2 weeks in soil, 2+ months in sea water, and there will result in exposure risks to eye, skin and respiratory irritation. Now, so under this research, they have proposed an alternative. What are the alternative? That is RNA interference. Here what they are going to do is, they are going to silence the genes, that is gene silencing techniques, targeting 4VPMT enzymes. Instead of an external output which is external input which is given to the locus, we are going to silence the gene itself with the RNA interference technology. Identified 7 safer molecular candidates for future research. The advantage is it encourages pollution-free locust, because you don't need 4NP for pollution-free locust control through biomolecular intervention. So what is the biomolecular intervention? Here is gene silencing of 4VPMT enzymes. If the 4VPMT enzyme is silenced, then it is not going to release 4VA. If the 4VA is not released, then it is not going to result in gregarious behavior or swarming. So, how do you do integrated locus control strategy proposed? So there is a strategy proposed, that is use synthetic 4VA analogous to trap and eliminate locust using fungi or limited pesticide. Spray 4VA disruptors to prevent swarming. Track population via 4VA markers and release genetically modified solitary phase locust in the wild. What they are going to do is, they will silence this 4VPMT1 gene in a particular locust, and then they are going to release this locust to the wild. When this locust mates with other locusts, it is going to produce an offspring where 4VPMT is already silenced. This is a technique which was previously used in dengue causing malaria, dengue or malaria causing female Anopheles mosquito. What they did was, they silenced a gene in a female Anopheles mosquito. The way that in the next generation it was not able to bear any offspring. As a result, the male population declined, and the male population declined. There will be no mating, and when there is no mating, obviously it is going to result in death of mosquitoes. That is being followed in locust as well. It combines molecular blockers with the biopesticides for sustainable control. Next, significance is, it marks a major breakthrough in eco-friendly pest control, reduces dependence on broad spectrum chemical insecticides, It offers an opportunity for precision agriculture and integrated pest management. So it is important for food security, especially in arid regions vulnerable to locust areas such as African countries, Pakistan, and northwestern India. So this discovery is a very significant discovery, not just from the science of molecular biology, it is also significant from food security as well. So that is why I covered this article or topic, a very interesting and intriguing topic. So at least just know what is the 4VA, what is locust warming, which are the areas affected in this areas, in this because of locust warming. Okay, so with this I will end the video. Jai Hind.